The life cycle of coronavirus. Here is a lung cell. Unbeknownst to this cell, it will be infected by a coronavirus and there will be no RNAi to save it in this origami demonstration. For simplification, the cell only has the Golgi body and the ribosomes depicted, which will be removed until they are necessary for further simplification. The first event in the process is the coronavirus phage coming in contact with the cell. The spike proteins, the orange tabs on the outside of the virus, attach to the host cell receptor, the blue tab, and enter the host cell. Here, a host of proteins on the surface of the virus aid in the process of invading the cell. Hemagglutinin esterase dimer, or the HE protein, aids this attachment process. The membrane protein, or the M protein, aids in the shaping of the envelope later. The envelope protein, or the E protein, is a very small protein which interacts with the M protein in the forming of the envelope. However, on this model, only the spike proteins are depicted. When the virus enters the cell, it releases its genome. Here, there are either two paths that the virus will take depending on external factors, which is a property of the coronavirus as well as other viruses. One is the lysogenic cycle, which will bring the genome of the virus into the nucleus and add it to the cell genome. When the cell reproduces as normal, it will make copies of its own genome along with the genome of the virus. The virus here is very sneaky as it can hide in the host cell genome for many generations. It stays with the cells until other external factors dictate whether the virus in the cell now moves to the lytic cycle. In the lytic cycle, the virus will reproduce many times within the cell until they break out of the cell, lysing it, hence the name lytic cycle. Keep in mind there will be much more than two viruses created in this process. In the lytic cycle, the viral genome is released, which is split into the antisense, or non-coding, and sense, or coding genomes. The sense genome is replicated as well as translated into a polyprotein and the viral protease. The viral protease is an enzyme to catalyze reactions, which, in this case, is to cut up the polyprotein created, which will form the replicase. The replicase, for all types of coronavirus, is now able to replicate RNA using the RNA template, which is the antisense genome from the original virus, like the transcription process. Through mRNA processing, the mRNA is shortened because the introns are removed, as well as other parts of the mRNA processing, such as the 5' cap addition, as well as any cleavage of the mRNA. Then, the mRNA leaves the nucleus and is translated by the ribosomes into the proteins known as the capsid, which will later encapsulate the viral DNA, which includes the spike proteins, which are represented by the orange tabs on the outside of the host cell. The coding genome of the virus, the sense genome, is encapsulated within the viral protein, or the capsid, which was translated from the mRNA. The entire protein DNA combo is then moved to the Golgi body, where they are shipped out of the cell within a vesicle. The vesicle from the Golgi body merges with the host cell membrane, and as the virus now passes through, effectively lysing the cell and creating the viruses. Keep in mind that millions of viruses can be created and the process will restart as the new viruses spread to other cells. And that is the complete life cycle of the coronavirus within a lung cell.